Welcome to the Penn Humanities Forum, this event in our ongoing series on peripheries. I'm Jim English. What? Huh? Sorry, I'm turning my phone off. I urge you to do the same. I'm Jim English. I'm the director of the Humanities Forum. I'm going to be super brief up here today because Professor Livingstone's talk is a little bit longer than our standard. She graciously offered to shorten it, but that would have meant losing the 3D glasses. I haven't been to every event the Humanities Forum has ever hosted in its 13-year history, but I believe this is the first time we've asked an audience to don the 3D glasses, so I didn't want to have that cut. But we will have a talk of about an hour, and then after that, as usual, we'll take a brief break of a minute or two, and then we'll have some question and answer time. I'm going to make one quick announcement, which is that the next event in our series, Junot Diaz, speaking about his journey from the peripheries of American letters to the center, was originally scheduled to speak on November 14th. He's so much at the center of the literary world right now that in addition to his MacArthur Genius Award that he just received, he's also been nominated for the National Book Award, and the ceremony is on the very evening that he was scheduled to be in this auditorium. So we've moved that to November 27th. Tuesday, not our usual Wednesday, Tuesday at 5 p.m. on the 27th. I encourage you to register for that event if you'd like to hear Junot Diaz, because we do expect a fairly full house for that. So that's it for my announcements. I will turn things over to Kevin Platt, professor in the Slavic Language and Literature Department, and he is the director of the Peripheries series, and he'll introduce our speaker today.
Thanks, Jim. And thank you all for coming. I think this is going to be a really fun and interesting lecture. Margaret Livingstone is a neurobiologist from the Harvard Medical School. And, you know, I wrote this and I said it, her work exemplifies how humanities can learn from the sciences and vice versa. But she said it's really the other way around. So note that down. Her main area of research is in the physiological processing of visual information, how our eyes and brains make sense of visual data. Yet she is a dedicated disciplinary border crosser, whose more popular book for both scientists and non-specialists alike, Vision and Art, The Biology of Seeing, brings the science of seeing to the experience of visual art. In a happy intersection with the Penn Humanities Forum annual topic for this year of peripheries, one of the many fascinating problems that Dr. Livingstone treats in her book is the curious role peripheral vision plays in the perception of works of art, including in the perception of the Mona Lisa's elusive, mysterious smile. Something that we might hear something about today, I hope. But if Dr. Livingstone chooses to tell us about other no less exciting matters, you can always purchase the book, which is about to be released in a new expanded edition. So please join me in welcoming Dr. Margaret Livingstone, who will speak to us on what art can tell us about how we see.
Actually, pretty soon not just how we see, but I hope pretty soon how we think. So as a visual neurophysiologist, I try to figure out how we see and how the brain works by recording from neurons in the brain. But I acknowledge that artists also try to figure out how we see, and many works of art are exciting or important because they reveal or discover important things about how we see. And for a lot longer than there have been neuroscientists or any kind of scientists at all, artists have realized that you can portray a scene by a line drawing. And it's so intuitive and so obvious that you have to stop and think for a minute that the scene that you're looking at does not have any lines in it. It's all contours and discontinuities, and yet you accept a line drawing as a perfectly good representation of a scene. And that is because the way your visual system handles the information that initially falls on the retina is not to record the amount of light at every point in the retina, but instead to record the relative amount of light at any point in the retina. I got this thing backwards again. So if you record from a retinal ganglion cell and shine a spot of light on the retina and record the signals from a retinal ganglion cell, you find, or Steve Kufler found, that you get a barrage of action potentials in response to a spot of light. Of course, the retina is for signaling light. But what he found that he was surprised at was that if you made the spot bigger, that is, you put more light on the retina, paradoxically, you get a weaker response. And Steve deduced that this is because retinal ganglion cells are excited by light at the center of where they care about in the visual field, but they're inhibited by light in the immediate surround. And you can see that if you shine an annulus of light on this retinal ganglion cell, you suppress the firing. So just in case you don't believe me that your visual system starts with this calculation, do you see the little gray spots at all the intersections everywhere where you're not looking? And don't worry about why it's where you're not looking, that's postgraduate. But do you see the little gray spots? So center surround cells at each intersection have 4 bits of white in their inhibitory surround, but center surround cells at the cross pieces have less white in their inhibitory surround. So the cells at the intersection are less excited, and so the intersections look less light. Because center surround requires an extra inhibitory synapse, there's a teeny delay between the onset of the center response and the surround, so you can actually see the dynamics of the center surround response everywhere, again, where you're not looking. Don't worry about that. So if you take anything away from what I tell you tonight, I hope you will remember that vision is information processing, not image transmission. It doesn't do you any good to send a high-resolution picture of the world to your visual cortex because there is nobody up there to look at an image. Vision is all about extracting information. All you have up there is nerve cells, and all they can do is fire or not fire. And these computations are simple, local, and often opponent. They're simple because all neurons can do is fire or not fire, and all they can do is excite or inhibit each other. That's a pretty simple calculation. They're local because they occur between neurons that are near each other in the cortex. Neurons are little tiny things. They don't project very far for the most part. And they are often opponent in that nearby neurons often inhibit each other. So why is center surround information processing? Well, I'm not doing too well with this. Well, that's because I'm pointing it the wrong way. If you have an image that looks like this, white on one side, dark on the other, center surround cells over here are not as excited because their surrounds are activated. Center surround cells over here are not activated because their centers aren't activated. It's only center surround cells at the border, at the discontinuity, where there's an imbalance between the center and the surround, that are excited. So center surround cells take a contour and turn it into a line drawing. And this is just to illustrate how informative and efficient a line drawing can be at conveying information, but it also illustrates this other fact. What color's the line? Black, right? This isn't hard, okay? Nothing is hard. The line is black. What color is the screen? White. Come on, it's white. So this is a nice projector, but it can't project black, right? All it can do is project light. So you know that the amount of light coming to your eye from the black line and from the white screen are the same. That tells you your visual system isn't interested in how much light there is at any point in the scene. It's interested in how much light there is relative to somewhere else in the scene. Because that's where the information is in an image. So not only throughout your visual system, in all kinds of computations that your visual system does, we use center surround opponency. And here I'll illustrate it in a number of different ways. We have center-surround opponency in contrast, so the checks in the center of both of these images are identical, but these look higher contrast because they're surrounded by lower contrast. Orientation is opponent, so the lines in the center are vertical, but they don't look vertical because they're surrounded by an off-vertical orientation. The red dot is moving vertically, but it doesn't look like it because of the opposing influence of the surrounding dots. Both of these X's are the same gray, but they don't look the same because they're surrounded by another color. The one on that side looks less blue because it's surrounded by yellow, and so on. And here's a little demonstration. If you look at that black dot, you will be looking at white letters, right? Keep fixating on the black dot. And white letters will give you a yellow after image because of the surround opponency in color. And of course artists know this. Monet painted blue shadows with little gold halos to illustrate the color opponency. So artists know something that even today's computer programmers are a bit off on, which is the way you can convey distance and depth in an image. So you can buy a very expensive computer program called a ray tracing program that will trace all the paths of light that will come into an image and reflect off three-dimensional things and make specular surfaces and shadows and stuff like that. And you don't have to do that. It's not necessary because the computations that are responsible for your ability to see distance and depth start out locally. By which I mean, early in the visual system, V1, primary visual cortex, each part of the cortex is concerned with a different part of the visual world, and the receptive fields are tiny. And this is where we first start our computations about distance and depth. So what do I mean by computations about distance and depth? You looked out at the world and it looks vividly three-dimensional, right? Come on, you have to agree with me or not. Yes. So the world looks three-dimensional. I know it is three-dimensional, I'm not denying that, but your visual perception that it is three-dimensional is carried by two flat retinal images. So how do you take two flat retinal images and get all this distance and depth? You do it by fairly complicated computations that involve relative motions. So when you move like this, things that are near move more than things that are far, shading, perspective, occlusion, things in front of each other, and stereopsis, which I'll talk about at the end. But all of these things start in early visual cortex where receptive fields are small. So it doesn't matter if an image has globally consistent depth cues, the way the computer graphics people think you have to have, and artists have realized or ignored this issue for a very long time. You don't have to test the rules of physics. You don't have to go around making sure the rules of physics are being obeyed. It's a given. So you can have paintings where the shadows go in two different intersecting directions, which makes no sense. You can have a vase in front of what appears to be a window with the shadow pointing towards the sun. This is at a museum at Harvard College. It's Canaletto's Piazza San Marco. And in this painting, the shadow, that's south, the shadows are going this direction, but over here the shadows are going this way. And when I pointed this out at the Fogg Museum, the curator said, oh, that's because the sun is being reflected from that building over there and causing those shadows over there. So I went to the Piazza San Marco. This building is full of windows. It does not reflect light, and people standing in the piazza do not have shadows going in two different directions. But nobody cares. Here's an Edward Hopper painting with sun streaming through a window, and that window ledge doesn't cast a shadow. Nobody cares. Here, I found this manger that is an impossible roof. You see the roof from the top on the left and from below on the right. But it looks three-dimensional. Obviously, Magritte was playing with this idea of occlusion cues not necessarily being consistent across an image. If you find any painting that's got a mirror in it, I guarantee you it is not obeying the laws of physics. if this lady were looking at herself in the mirror, you, the viewer, would not see her face, right? Right. Even Vermeer painted this nice shiny brass bowl, and it looks shiny because it's reflecting the tablecloth. But if you look carefully, what is reflected in the bowl has nothing to do with the tablecloth that's underneath it. So something else that artists figured out way before us neuroscientists is that luminance, and don't worry, I'll define this in a second, luminance and color do different things for you and can be treated differently when you're trying to make a work of art. So we have 3 cone types in our retinas: short, middle, and long wavelength, or blue, green, and red, loosely speaking. We see color by comparing the activity in pairs of these cones. We see luminance just by summing them up, like black and white photography. And in this image, color on the left, luminance on the right. Most color contrast borders also have a luminance contrast. Oh, for those of you in the art field, when I say luminance, I mean value, and I'll try to remember to say that, but you may have to translate for me, okay? So most color contrast borders also have a luminance or value contrast, but you can have color contrast with no luminance contrast, and when you do, rather peculiar things happen. And one of my favorite illustrations of this is Monet's Impression Sunrise, the painting after which the Impressionist movement got its name. The sun in this painting, and you owe it to yourself to go to Paris and see it, is absolutely brilliant. It seems to shimmer. It kind of floats in a weird way, and it shivers and looks hot and cold at the same time. And I once got a very small grant from Harvard to go to Paris to measure the luminance in Impression Sunrise. And it turns out, despite how bright the sun looks, it's actually exactly the same luminance as the off-white, as the gray background. And I think it's actually because it's not brighter or darker than the sky that it has this weird shimmery quality. And that comes from the fact that your visual system, and the primate visual system in general, is subdivided into these 2 major streams. And with information coming in the eye to the primary visual cortex, and then splitting into these 2 divergent pathways that do very different things. And we know about what they do because of what happens when people have strokes or gunshot wounds or something to that part of their visual system. So the more ventral stream, the one in the temporal lobe, seems to be important for object recognition because people who have lesions in this part of their visual system develop what are called agnosias. They can fail to recognize very particular and sometimes quite peculiar categories of objects, like faces, or fruits and vegetables, or writing, or color. So we call this the what system because it seems to tell us what objects are out in the world. This part of our visual system is evolutionarily new. It's present mostly only in primates. The other part of our visual system, the dorsal stream, we share with other mammals. And this part of our visual system does what you'd expect a self-respecting visual system to do for an animal that needs to navigate through its environment and tell when something's moving because it might be predator and it might be prey. Some people call it the where system because cells there carry information about motion, depth, and spatial organization, and some people call it the how system because it's maybe information that we use for interacting physically with our environment. So here are some drawings made by people with lesions in their parietal lobe. These patients were asked to draw a bicycle, a cross, and a clock. So you can see that these patients got all the bits right, but what they're missing is the spatial organization of all the bits. They almost look like cubist drawings, right? Not to say that Picasso had a parietal lobe lesion, but I think Picasso cottoned on to the idea that what something is and where it is are 2 quite distinct things. But what's important for what I want to talk about now is that, like other mammals, this evolutionarily old part of our visual system is colorblind. So if you had a red spot on a green background and you saw it with your WARE system, or you took a black and white photograph of it, you would get a gray spot on a gray background, right? You can imagine changing the relative brightness of the red in such a way that you got the same grays. That's equal luminance, and very peculiar things happen at equal luminance. So you can see that some of these gray spots have this weird shimmery quality, like the sun in Impression Sunrise. I think that's because there's a whole part of your visual system that can't see them. So now I am going to try to convince you that your ability to see motion, your ability to see depth, spatial organization, and figure ground segregation are carried by a colorblind part of your visual system. So I'm going to show you, so we may need to lower the lights for this one, okay? So in red, so these are some moving dots, okay, right? And it looks three-dimensional, I hope, because of the relative movement of the red dots on the green background. The direction that the dots are moving is ambiguous. They will probably reverse at some point. Don't worry about it. It's not interesting. So now I'm going to try and make the red darker than the green, and it still looks three-dimensional. And if I make the red brighter than the green, it looks three-dimensional. But if I go through equal luminance, I hope you find a point where this gets all flat and squishy. And then it gets three-dimensional again. Let's go back through it. Whoops, I went too fast. Flat and squishy, right? And then it gets three-dimensional. I could do that all day, but I won't. And if I hit escape, I can get in trouble. No? Oh, okay, good. Ha. Now, I bet you can see that this has a lot of cues about shading and perspective, but it may not look as three-dimensional until you close one eye and look through the red lens of the glasses. And then I hope it looks more three-dimensional. Okay, and then I'm going to take off this thing that's making noises here. There we go. All right, so now put the glasses down, please. Thank you. Now, but first of all, look at this and see if you can see the depth from perspective, the depth from shading, and even the figure ground segregation. Can you figure out what is doing which in this image? And then close one eye and look through the red lens and see if it doesn't look more three-dimensional, and if it doesn't make more sense, if you don't get figure ground segregation and all kinds of figuring out what this image does. Okay, glasses down. You can leave them on now if you want, but they aren't going to do you any good. Monet painted a bunch of versions of this cathedral in Rouen. Because it's just a beige stone building, it's all about light, shape from shading and depth from shading. And you can see the depth in the doorway and the rose window, and it's all there in the luminance version alone. But some of them look flat and shimmery. The building faces west. This is the cathedral at Sunset. This one's at the MFA in Boston. And it looks flat and shimmery because he's using just about equal value, equal luminant colors. And no glasses. Thank you. Now, without the glasses, think about, do you see anything in this image? This is Liechtenstein's series, part of Liechtenstein's series of the Rouen Cathedral, and I bet it doesn't look like anything until you close one eye and look through the red lens. The red lens blocks the blue light, okay? So you introduce a luminance contrast. One eye, close, just look through the red lens, right? Okay. So I hope I've convinced you that your ability to see depth from shading and perspective are carried by a colorblind part of your visual system because you require a luminance contrast. The corollary is that it doesn't matter what the colors are as long as the luminance is appropriate for you to see the three-dimensional shape of something. And I think some artists figured this out already. This is André Derain's portrait of Matisse. If you knew Matisse, you would recognize him because even though the colors are very peculiar, the luminance is exactly right for conveying the shape of his face. And recognizing a shape of a face is a very subtle thing to do. And in Matisse's Lady in a Hat, again, the planes of her face are bizarre colors, and everybody got really upset when he came up with this painting. But he's using colors symbolically, like Picasso said, and the reality is there in the luminance alone. So I hope I've convinced you that your ability to see depth, spatial organization, and figure ground segregation are colorblind. Now I'm going to try to convince you that your ability to see motion is colorblind. And I'm going to do it again with a demo here. And I have to slow it down first just a little bit. So you can cheat if you want. I wish you wouldn't, but you can. And look at this side of the image. And what I want you to notice is whether the two sets of stripes are moving at the same speed, and whether the green is brighter than the red or darker than the red. They should be seeming about the same speed, but when I go through equal luminance and you're not looking at the center and cheating, I hope you notice that the one on the left seems to be going a lot slower because it is a suboptimal stimulus to your motion system. So if your ability to see motion is colorblind, your ability to see that something is stationary is also colorblind. And I think Monet achieved a sense of motion in many of his paintings by using equal value, equal luminant colors. So the poppies are just about equal luminant with the grass. Your what system can see them, your where system can't, so their position can be indeterminate. They can seem to move in the breeze. I don't know if Monet thought he was managing to achieve a sense of motion in his paintings, although the art historians all think he did do that. But I know Mondrian knew that he had achieved a sense of motion in this painting because he titled it Broadway Boogie Woogie. The original is at the MoMA in New York. It's about this big across, and it is absolutely nauseating the way it jumps around. And I think it jumps around because your what system can see the yellow checks, but your where system can't, so their position can seem to change. Advertisers do this all the time. I bet you if you find a Wired magazine somewhere, you can find an ad that's got one color against an equal luminant background, and it's attention-getting because it jumps around, and it's has the benefit to the advertiser that it's harder to read, which I will get to at the end, I hope. So I hope I've convinced you that your ability to see motion, depth, spatial organization, and figure ground segregation are both colorblind and start locally with their computations. Now I want to try to convince you that your color perception is coarse or low acuity, because at any point in the visual field, the cells that carry information about color with their co-opponent receptive fields tend to have large, low acuity receptive fields. So you do not have to color inside the lines. Of course, artists know that. Pastel artists, watercolor artists put on an outline and slap some color nearby the line, and it conforms to the line. Here's an illusion based on this. I hope you see the donut as yellower than the whole or the background. You have to agree? Yep, you do. Okay, good. Me too. But I know, despite what I see, that the donut is exactly the same white as the whole and the background. This is a purple line with a yellow line inside it. Because your color acuity is so low, your visual system lets color spread till it hits a border. Now I want to show you using after images, which happen when your photoreceptors run out of energy. I want to use after images to show you color spreading in your visual system until it hits a border. What I want you to do is to look at the intersection right between these three shapes, okay? And if you look at the intersection, then I'm going to show you an achromatic gray background, and you're going to see a bit of an after image. Now I want you to do it again. Look at the intersection and see if you don't get a better after image in this achromatic version. Yeah, you just figured out it's achromatic, didn't you? Right. And some of you sophisticated people in the audience are saying, well, of course it was better the second time you did it twice, so you built it up. No. Look at the intersection, evaluate the after image. Look at the intersection, evaluate the after image. Now here's another version. Look at the intersection and see if you can get that color to fill a completely different shape. And look at the intersection and now watch the colors squirt out the hole. This is a very famous painting. I know you've seen it hundreds of times in advertisements and with mustaches and postcards and things. But there's a reason why people love this painting so much and why you can't get near it at the Louvre. the Mona Lisa looks almost alive. Her expression seems to change as you look at it. And I was reading this art history book about this, and I realized that the art historian said that her expression changes because her smile is ambiguous, because Leonardo used sfumato. But I realized that when I looked at her eyes and thought about how much her mouth was smiling, and then looked at her mouth, she seemed to be smiling more when I was looking at her eyes than when I was looking directly at her mouth. Did you see that? Her expression varies systematically depending on where you're looking. That's not ambiguity. That has something to do with really low-level vision, right? So your visual acuity falls off dramatically from your center of gaze. So if you look at that dot in the center, all of those letters are equally readable. Your central vision is really good at seeing tiny, highly detailed things. That's why you have to move your eyes when you read. You can't see those little tiny letters with anything except your center of gaze. But your peripheral vision isn't bad, it's just different. It's specialized for seeing bigger, blurrier things that your central vision can't see. So if you filter the Mona Lisa in such a way that you'd see what she looked like to your peripheral vision, if you could see the whole thing with just your peripheral vision, or your central vision, if you could see the whole thing with your central vision all at once, which of course you can't. And of course she looks much cheerier in the low spatial frequency cuts, that is to your peripheral vision, than she does to your central vision. You move your eyes constantly. I'm going to talk about this in a minute. You can't stop moving your eyes, and if you do stop moving your eyes, you stop seeing. So it's absolutely essential that you move your eyes. You make tiny eye movements all over the place several times a second. So as you move your eyes, her expression changes, which gives this painting 500 years ago a dynamic quality, which was pretty special 500 years ago. I think it also gives her a coy quality. So you notice when you're looking at the background that she seems to be smiling, and you try to look at her real fast and she stops. The post-impressionists, the pointillists, said that they achieved a dynamic, vibrant quality to their paintings because they made the colors out of little dots of pure color. And they thought that the vibrancy had to do with additive color mixing. That is, you're adding the colors separately, not smearing them together on the canvas. But magazine illustrations are made up of additive color mixing, and there's nothing special about the colors in a magazine, right? So I don't think it's the additive color mixing that makes these paintings sparkle. I think it's the fact that with your central vision, you see the dots. With your peripheral vision, you see the river and the trees and the boats. And as you move your eyes around, it changes. And I think that's what makes it vibrant and dynamic. Chuck Close makes portraits out of these giant tiles, and you can't even see what the painting's about until you manage to look at it either far away or with your peripheral vision. And photomosaics. With your central vision, you see baseball cards. With your peripheral vision, you see Babe Ruth. So now I want to talk about how we recognize objects with this primate-specific, object-selective part of our brain. And I'm going to talk about processing faces because we, primates, are very good at recognizing faces. These images are going by in 1/10 of a second each, and yet you are recognizing faces, detecting faces, and actually recognizing individuals, even when they're degraded or occluded or disguised. And you are better at this than any computer program. So how we recognize faces, again, the artists were way ahead of us on this. You find it much easier to recognize a caricature of an individual than to recognize a veridical line drawing. Artists have been making caricatures for 2,000 years. Why do caricatures evoke an identity so much better than a real representative image? So that turns out to be something that the artists have recognized about face processing. We've studied face processing in monkeys who also have so people have localized regions of their brains dedicated to processing faces. We know this because you can have a lesion that specifically wipes out your ability to recognize faces, and because you can see it on functional MRI. You can also see it on fMRI in monkeys. Monkeys, like people, use faces to recognize conspecifics and to convey important social information. So we designed, so the face cells in monkeys respond to monkey faces, they respond to people faces, and they respond to cartoon faces. So we used cartoon faces to say, what do face cells care about? How do we recognize other individuals? And we designed this cartoon face space where we varied 20 different parameters about faces, eye shape, nose shape, mouth face shape, everything we could think of. And we showed this stimulus to cells and said, so we're varying all of these things simultaneously, and we said, does a given face cell want one of those images, or does it care about these parameters that we're varying systematically? And the answer was that cells cared about parameters. And interestingly to us, it's a bit of a detail here, is that each face cell cared about 2 or 3 of these parameters. It didn't care about the whole face, it just cared about inner eye distance or mouth shape or something like that. But more interestingly was the way these cells were tuned to these parameters. So if you had a cell that cared about inter-eye distance, you might expect to find, these are supposed to be 6 different cells, each of which are peaking in their selectivity to a particular plausible inter-eye distance. That's sort of what you'd expect to find, but that's another way to do the same thing, to code inter-eye distance, would be to have one cell that liked really far apart eyes and one cell that liked really close together eyes, and then all you need to get all the eyes distances in between is the ratio of the 2. And it turns out that's what face cells do. So this is the tuning to all 20 parameters of one face cell, and each of these little graphs shows that of the parameters that this cell cared about, they were all ramp-shaped. They were tuned to extremes. And you saw that those were weird-looking faces, right? We went pretty far extreme, thinking that we were going to get outside face space, but we didn't. But in any case, it turned out to be the case that face cells are tuned to these variables that describe faces, but they're extreme. They're excited by one extreme and inhibited by the other, or vice versa. Ramp-shaped or extreme tuning is consistent with the idea of why caricatures should work. What is a caricature? You take somebody's face and you say, how does it differ from the average face? And then you exaggerate the differences. So that's what face cells do. They say, how does a face differ from the average face? So that and it also means that face processing involves comparisons of one face to another, and it pulls face processing into what I've been telling you about all the rest of the visual system at lower levels, which is that it's local, that is, between faces in face space, and opponent. So you may have seen many Picasso portraits, but I bet you never looked at the portraits next to a photograph of the person that was a portrait of. And what's fun is that they're caricatures, aren't they? That's Gertrude Stein at the top, Picasso, the wife of a friend, his art dealer, some guy he roomed with, Igor Stravinsky, wife, girlfriend, mother-in-law. They're all caricatures, right? He's managed to capture what makes each of those faces distinct. Now, if face processing, now while I'm nattering on, I want you to look at these little numbers in the center, okay? Because what I'm going to try and do is adapt you to these 2 faces. So if face processing is local and opponent, then in the same way that you can get color after images, you ought to be able to get a face identity after image. And you're moving your eyes so that we're not getting just a retinal after image. We're getting a real high-level identity after image. And now you're going to see 2 identical morphs. But I hope you saw a face identity after image. So I have been trying to convey the idea that vision is information processing, not image transmission, and I've showed you many ways in which it is information processing, but you can look at any work of art and see information processing. you can see information processing in a child's work of art. You ask a 3-year-old child to draw a person, and you will get a face on legs, right? Any 3-year-old child. The child is telling you what's important about a person. What is the information that child has registered and is now feeding back to you? That the face is the most important part about a person. That's another 3-year-old. This is a picture a 3-year-old child drew of his pregnant mother. A face on legs inside of a face on legs. Matisse recognized the opponency of the information processing explicitly. He says he doesn't paint things, he paints the differences between things. So I got invited to be on a panel at the Museum of Art and Design in New York a while ago, and the panel was about dioramas. And the last time I had thought about dioramas was when my son was in the 5th grade, with the shoe boxes, right? Remember those? But dioramas are kind of a thing now in the art world, and they are exquisite. I was quite surprised by how interesting they were. Why should looking at something at a very different scale make you see it differently? And I think artists do this a lot. When you get outside of early visual cortex into object selective cortex, you no longer have tiny receptive fields. You have cells that care about big regions of receptive fields, but what they care about is particular categories of objects. So they'll care about faces or body parts or something quite specific. And I think this modular organization of the temporal lobe is something that artists were tapping into when they did things like making dioramas. So you have surprisingly specific kinds of agnosias, which means you can have surprisingly specific cortical modules for recognizing particular things. Small objects, big objects, places, faces, animate objects, writing, body parts. Those are all the different kinds of agnosias you can have, which means that those are also the kinds of modules we probably have in our temporal lobe. So work that's going on in my lab right now suggests that these modules are not innate, but they rather develop because we learn, our visual systems learn to get good at recognizing things we encounter a lot. And the way you get good at something is by making a little module so that cells nearby can be dealing with the same object and can interact with each other. That's what makes you good at faces. You have within category comparisons. So I'm going to illustrate to you how good you are, how differently it is when you look at something with your face module compared to when you look at it with ordinary non-face cortex. So because you grow up seeing faces most of the time right side up, your face module is built up of cells that are selective for upright faces. Now you know that this is the Margaret Thatcher illusion. You may not know that's what it's called, but you know that I have manipulated one of these images, and you know that one of them is going to look weird, right? You're seeing this face, even though you know it's weird, okay, with non-face parts of your brain. So you're processing it with non-face parts of your brain. I am now going to turn it over, and you're going to process it with your expert face processing part of your brain. Even though you were prepared for this, you see it differently, right? So that shows you how differently you see things when you're using an expert part of your brain as opposed to a non-expert or general purpose part of your object recognition system. And I think artists figured this out. And when they make things that are the wrong size, for example, I think you process it with a different part of your brain and look at it differently and can see things that ordinarily would look not the least bit interesting are interesting. Or even, not even making it a different scale, but putting it in the wrong place. So now again, you know there's something, does it look hideous again? Right, isn't that weird? It's just astonishing to me how different things look when you process it with one part of your brain versus another. So now I want to talk about the idea that your perception of the world is continuous. You look out on the world and it seems to be nice and stable and it's not jumping around, and I know it's not jumping around again, but You're figuring this out with retinal images that are constantly jumping around. So if we could turn the lights up just a little bit, I want everybody to close one eye and then gently tap on the lid of the other eye. Don't poke your eye, just tap on the eye. And notice how the world jumps around when you do that, okay? It's very unpleasant. Don't poke your eye, okay? Yet you move your eyes continuously. So when you know you're moving your eyes, when you have some kind of motor command to move your eyes, the world doesn't jump around. But if you move your eye, the world jumps around. And here's just an illustration of how much you move your eyes. Don't worry about why this is working. This is a static image, and yet every time you move your eyes involuntarily, you see the globe thing shifting, right? Remember I told you that if you don't move your eyes, vision fades. So there's this purple spot up there, right? But if you look carefully at that X, I bet you will, if you're really good at fixating, I bet you'll see that thing fade, right? You need to have, so your visual system, in the same way that your visual system is interested in discontinuities, because that's where the information is, your visual system is also interested in things that change. And things that don't change, like rocks, are not interesting. So if you look at the P up there rather fixedly, if you can do that, you will notice that the whole thing fades to pink. And now look down at the B, and now the whole thing will fade to blue. Isn't that peculiar when it does that? I think a number of artists figured this out way before us neuroscientists. Turner, for example, you find a Turner painting and you look at it and it's so blurry and fuzzy in the rest of the image that you'll find fading happening, which is kind of disconcerting. And I think Mark Rothko cottoned onto this too, because his paintings have blurry edges and they're supposed to be placed in rooms that are quite small so that you need to be very close to them. So they fill your visual field and the edges are blurry and they can seem to fade to whatever color's on the outside, which is disconcerting, or, as he puts it, mystical. So I think that filmmakers are way ahead of us neuroscientists in understanding how we take these constantly changing retinal images and convert it to a perception of stable reality. Filmmakers know that when movement happens in a scene, you move your eyes, and then you can change a lot of things in the image, like do cuts, and you don't find it disturbing. So here is a demonstration of how moving your eyes can make motion seem to disappear. So what I want you to do is see that little red dot at the bottom, track it, okay? And while you move your eyes, if you track that really well, I hope you will notice that the bulging red dots seem to stop moving as you move your eyes. Does everybody see that? It's pretty good, huh? I'm gonna have to get this off of here somehow. So, one fact then that we can deduce is that if you move your eyes, your visual system doesn't worry about things changing. And obviously filmmakers take advantage of that. What have I done? Oh, I've gotten, why don't I just get rid of this and stop confusing me here? Oh, what did I do? So here's a tutorial
Here are two
on
shots.
film cutting.
One wide and one close up. Cutting on motion allows you to go from a wide shot to a close-up in what will appear to be a seamless edit.
Wasn't that beautiful? So, sorry, I think it's really impressive. So the visual system ignores changes that occur during eye movements, but then we have to link one frame to a completely shifted frame. Many neuroscientists think that you do that by remapping the retinal image. So, for example, if you're fixating at the center of this grid and there's something over there that you want to look at, your visual system somehow shifts the coordinate system. And there are actually these unbelievably complicated models out there for how we do that. But I think filmmakers are way ahead of us and have realized that there's no way that we're doing some complex calculus-based remapping, because you can go from a wide-angle shot to a close-up, and there's no way that your visual system was designed to do that, because we don't have a zoom on our visual system. So I think we just ignore movements that happen when we shift our gaze, and then we just link together one frame to another frame. And what we use to link together one frame to another frame is either common motion or common objects, or both. But because we don't have to go around checking that something that's hurtling across the room is the same object from one side to the next, we sort of assume it is, that you can have very strange things happen and it doesn't bother you. And I think filmmakers know that. This is the drain in Psycho, Alfred Hitchcock's drain in Psycho. And this is 2001, A Space Odyssey, with the bone that turns into the spaceship. And here is a series of cuts of Jim Carrey movies where you have seamless transitions from completely unrelated scenes because of the commonality of movement and gesture. And this is less disturbing than moving your eye, right? You see this continuity. Sorry, I had to do that. Now, the last thing I want to talk about is stereopsis. So, look through your glasses, both eyes open this time, blue on the right, okay? For those of you who have stereopsis, and don't worry if you don't, okay? For those of you who have stereopsis, this thing will look more three-dimensional. Stereopsis is based on the difference in the images in the two eyes. So if you're looking at the dots cast images on corresponding parts of your two retina. You don't need your glasses for this. But images that are farther away or closer cast images on non-corresponding parts of your retina, and your visual system uses this difference in the two images as part of its computation of distance and depth. So if you go to a museum or find a painting and you're looking at it and it's got shadows and perspective and occlusion cues that there's a lot of depth, and you're standing in front of it with two eyes open, your stereo system says, no, I'm sorry, it's flat. Maybe a nice painting, but it's flat. If you close one eye, when you can find a painting like this that fills your visual field, you will find that you almost fall into the painting after a few minutes. It looks more three-dimensional when you close one eye because your stereo system is not contradicting all of the nice depth cues that the artist put there. But stereo matching is computationally a very difficult problem. So now, no glasses, okay? If I tell you, and you're a smart audience, that the cyan squares are white to your right eye and the red squares are white to your left eye, can you figure out what this shows? That's a simple algorithm, right? All right, see if your visual system can do it. Blue on the right, red on the left. Some of you will see something. Some of you will not. Don't worry about it. See? So that your visual system solved this computationally very complex problem that you couldn't do in a million years. But stereo matching is a difficult problem. If you have 4 identical images in the real world, you have 4 images on each retina, but you could match them up correspondingly, but you could also match them up non-correspondingly, which would correspond to the same object, but at a different distance or depth. So if you've ever misstepped on an escalator, you did that because escalators have these stupid slats that can be mismatched in your two eyes, which would correspond to the stair being in a different place than you think it is. I think the Impressionists and the Post-Impressionists, when they said they were painting the air, did so. So these are paintings that after you look at them for a while with both eyes open, you get a lot of mismatches in your two eyes, and I think that corresponds to kind of a speckly volume of something or another. And this one, I don't even like to look at this one because it sort of undulates as you mismatch the leaves from one part of your visual field to another. But what's peculiar about this painting is that Klimt, who made it, could not possibly have had stereopsis. And how do I know that? He's cross-eyed, right? He's severely cross-eyed. I told you that the computation starts in primary visual cortex, where the receptive fields are small and the map is retinotopic across the cortex. So if you are cross-eyed, you end up with non-corresponding parts of the visual field in the same part of the cortex, and you can't possibly compute stereopsis from that. I got interested in this when I did some research on dyslexia, which I started doing because I noticed that it's really hard to read equiluminant writing. And people with dyslexia is a selective problem in learning to read for people who are otherwise of normal intelligence, motivation, and education. There are quite normal people who have a terrible time learning to read. And they complain that text seems to shimmer or jump around on the page, just like what I find equiluminant text does. And so I did some experiments which led us to the idea that people with dyslexia have a slight timing distance in their wear system, surprisingly. I think possibly because reading involves a lot of fast eye movements and you have to link one part of the text to the next, and they may have trouble doing that. That may be the shimmering that they see on the page. In any case, if there's something, remember what the wear system does: motion, depth, spatial organization, and figure ground segregation. Well, what are the other symptoms in dyslexia besides a selective problem learning to read? Well, positional information, figure ground problems, and trouble gauging distance and depth. So in the course of this research, I was privileged to meet a large number of people with dyslexia, most of whom thought that this was a kind of an interesting idea. But I noticed that most of these people were very talented artists, musicians, computer programmers, or actors. And it's well known that dyslexics are overrepresented in the artistic population, but the usual assumption is that they were bad at academic subjects, so they spent most of their time in the art room. But the talent that I observed in some of these people that I met made me consider the idea that maybe there's something about dyslexia that makes them better at art. So there's auditory problems too in dyslexia. That's why they have trouble with phonetics, because the difference between ba and da turns out to be a very fast discrimination. And so maybe there are many more people with dyslexia in music because they may be bad at fast transitions, but maybe they're better at slow auditory transitions. So why are there so many artists who are dyslexic? Well, maybe trouble gauging distance and depth could be an asset to an artist. If I try to draw a chair, I can't do it, right? I am stuck with the information that this thing is three-dimensional, and I have no access to the early level flat retinal images. I've extracted the information and I don't have access to it. But if a kid sees things as flatter than his friends do, maybe it's easier for him to draw it on a flat piece of paper, and just a little bit of a head start as an artist, as a kid, and get you interested in it. And so, in any case, this is Chuck Close. Severely dyslexic, says he sees things as flat. He has bad vision in one eye. So there's a test. You can tell whether somebody's eyes are lined up by where the light reflection, those little white spots are in the two eyes. So if your eyes are not lined up, you can't have good stereopsis. If they are lined up, you could. So 10% of otherwise normal people have no stereopsis. But this is called the Hirschberg test. It's actually a clinical test that you can use, is whether the light reflexes in the two eye are in the same place. So here's a couple of baseball players. Do you see that the light reflexes are in the same place in their two eyes? Okay, are you good at this now? You can do this test? Okay, there's one baseball player who is known to have had no stereopsis. They root. See the light reflexes on half the eye on the left and right in the middle on my side. He had amblyopia. He was essentially blind in one eye. So when the images from the two eyes come into visual cortex, the visual cortex can either alternate which one it looks through, or it can ignore the input from one eye, in which case essentially the connections from that eye wither away and you lose the functionality of that eye. That's called amblyopia or lazy eye. So only 3% of the population has really misaligned eyes. Even though 10% of the population is stereo blind, only 3% is stereo blind because their eyes are all misaligned. So we got some photographs of famous artists and said, there was like 60 of them that we got from the National Portrait Gallery, and we said, how many of these had misaligned eyes? You'd expect to find one or two, right? We found 16 right off the bat. This is Andrew Wyeth. That eye is looking at you. This eye is looking over there. His father, N.C. Wyeth, who was also an artist, was also strabismic. Edward Hopper, you see the light reflexes in two completely different places in the two eyes. Kara Walker, every picture I've ever seen of her, the light reflexes in two different places on her two eyes, and she works in silhouettes. How flat can you get? I don't know about the dog. Marc Chagall was cross-eyed. Frank Stella was cross-eyed. Jasper Johns was wall-eyed. His eyes deviated outwards. Robert Rauschenberg was wall-eyed and severely dyslexic. Alexander Calder was wall-eyed. Man Ray was wall-eyed. Frank Lloyd Wright was wall-eyed. Now, I told you that it is an accepted clinical test to test the alignment of somebody's eyes with a photograph. I know you're not supposed to do this with a painting, but at the Louvre, there's a little room that all it's got in it is 4 Rembrandt self-portraits, one on each wall. And I couldn't help but noticing that in every single one of them, he paints himself as having one eye looking straight ahead and the other eye looking off somewhere else. Now, I know when you make a self-portrait, you use a mirror. And you look to the right when you're painting your right eye, and you look to the left when you're painting your left eye. So here's a montage I made of myself with a mirror like 8 inches from my face, with that eye, this half looking at my right eye, this half looking at my left eye. So you can see, even though I have normal stereopsis, the light reflex is not in exactly the same place in my two eyes. But my eyes are nowhere near as deviated out as Rembrandt portrays himself. So at this point, I had piles of reproductions of Rembrandt self-portraits on my desk, and my young colleague came in and said, my young colleague, Bevel Conway, who is himself a stereo-blind artist, said, "Is it always the same eye?" And I said, "No, if it were, I could publish it." Sometimes it's the eye on the far side that deviates out, and sometimes it's the eye over here that deviates out. And Bevel, being a stereo-blind artist, said, "Marge, you have to separate the paintings and the etchings." So when we did that, in the paintings, it was always the eye on this side that deviates out. In the etchings, it was always the eye on this side that deviates out. And of course, you make an etching by scratching on a plate and flipping it over, right? So we did a little a Photoshop program where we outlined the eye and figured out where the iris was on both sides, and then we plotted a graph. Take-home message is that if you can make a graph of the unlikeliest thing, you can get it published. And in the paintings, it was always the eye on the left that deviated out, except for this one, which is in Berlin, and we think either it's a fake or he painted it from an etching. And it was in general the reverse for the etchings. So we think, so if it were a trope, do you like that word? If it were a trope, then he should have had the etchings and the paintings all come out the same, right? Because he used to repaint himself so that he didn't look like he was left-handed when he used a mirror to paint himself. So if it were a trope, he would have reversed it. The fact that it's mirror image reversed in the paintings and the etchings suggests to us that he really was stereo blind, which you don't have to, so if you want your kid to be an artist, you don't have to go around poking out an eye. One of the first things you learn in art school is how to flatten the world, and one of the first techniques they teach you is close one eye, flatten the world, or look at it upside down, or look at it in a mirror. So, if you want to have some messages to take home, the most important one is that vision is information processing, and the one that I like is that people are different, and some things that may be good for one job may be bad for another. And I'm happy to take questions.
Yes. Did I understand you correctly that Babe Ruth was blind in one eye?
He was essentially blind in one eye. And what I find interesting about I showed a few baseball players as examples of people with good stereopsis. So nowadays you can't get in the major leagues unless you have perfect vision in both eyes. But that's honestly really unnecessary because the baseball comes so fast that you don't use stereopsis to hit it. You plan, as I understand it, you plan your swing based on what the pitcher looks like when he lets go of the ball. You can't use stereopsis. Stereopsis is only good within like 10 feet because any farther away, the two images are virtually identical in the two eyes. And so I suspect Babe Ruth had hyperacuity in his good eye. And so that he could actually see better with one eye far away than we could with two eyes. And that's why I suspect, and there'll never be another Babe Ruth because they wouldn't let him in now.
Amazing. Thank you.
Yeah, thank you. I had a question. It sort of came to me when you were showing the film sequence. And
The what?
The film sequence, the rapid cutting film sequence.
Oh, yeah.
That you showed.
The Jim Carrey, right?
And that was at the point where we were talking about motion. But what happens with sound? Because for me, it was really the sound that was creating that suture. So how do you relate the sound? And then when you were showing your slides about dyslexia, it was also, there's an acoustic component to the dyslexia. So I was wondering if you've done any
You're right.
research on how the sound then goes into that field of motion.
So first of all, I haven't done any research on this at all. I work on low-level
Sorry.
vision. This is a hobby.
Okay, sorry.
But yes, you're probably right, and I ought to think about that. But I work on vision, so I never think about sound. But you're right, I've heard some fascinating, I've been paying attention to cuts recently in movies, and you're right, there's some of them that go from thunder, that there's a continuity of the sound that's very interesting. You're right.
Thank you for your talk. Is this one? You think? Another sport and vision. This is why I love the talk. I've noticed that when there is a major of the tennis, there are 4 majors, they have a draw of, let's say, 128 people. You usually have the same people, the top 10 are the top 10. And then you tune in, and there they are. The top 4 are always in the semifinals and then the finals. So I listened to sports commentators, and they said, well, why are the top 10 in the top 10? And they would say it's desire, who wants it the most, etc., and being well prepared, and their fabulous training. And I started noticing things about their eyes, that the top 5, the top 10, they have a type of a Cro-Magnon caveman forehead of the eyes are indented way in. Federer is an example. Murray is an example. And then they're close to the bridge of the nose. It never misses. The top 4, 5, 10 have those eyes. And they say about your ball coming out of the pitcher's glove that the success of Jimmy Connors, and he played professionally until 39 years old, they said he had a phenomenal gift of knowing where the tennis ball was going to go as soon as it left the racket of the opponent. So I'm giving you a homework to research that.
Well, the brow is associated with testosterone, so there may be other things going on there.
I came late, so perhaps you discussed this already, but what about art cultures that don't have perspective, that don't use perspective?
So one of the things I took out of this talk was that there are many works of art that reveal important things about human culture or human psychology. And mostly I don't talk about those because I work on low-level vision. But you're right that Eastern paintings don't use perspective in the same way we do. But I don't think that means that people in those cultures see perspective any different than we do, because it's just a function of, it's just that it wasn't something that evolved in their art. Because art isn't, the goal is only, for a short period of time, the goal was to make highly representational, three-dimensional paintings that you could look through a peephole and think you were in the Piazza San Marco. Before that, it was all about people being illiterate and telling stories of the Bible, and it was a big part of the church. And it was probably equally a big part of culture and conveying stories for a long time in Eastern paintings too, not making something that looked like a scene in the real world. You have to do the microphone.
Shall I ask?
Yeah, over on this side?
Yeah. Thank you. This was exhilarating. I work with color. I do interior design. I'd love to understand more about what you said about color perception being coarse.
Oh.
Did I misunderstand it?
No, you're absolutely right. Guy would just go look at a watercolor. I mean, artists have figured out you can do this. So television, do you remember when I was a kid, they invented television, and it was black and white. And everybody who wanted a TV station got 6 megahertz of bandwidth. And then CBS wanted to make a color TV, and they tried to get 18 megahertz of bandwidth, and they couldn't. And so they had to make a teeny TV. Nobody bought it. But then RCA came up with a solution that lasted until about 5 years ago, which was to put most of the signal in a high definition luminance signal and then a really blurry color signal on top of that, and nobody cared. It's just something you don't notice. So most of the time, color is associated with a luminance border, but if it isn't, you don't care.
That's why I have so many problems with my clients.
Yeah, well, so most men think that they can tell the pixel-wide resolution on these high-density TVs. It's completely unnecessary. I don't know why they do it. It's a guy thing.
Not me. I had a question about, it's a sort of follow-up on the previous question. It's about the cultural specificity of different kinds of perception effects. I was also struck by this when we were looking at film. Obviously, there are certain things which are learned behaviors, learning to recognize specific objects or specific faces. And then obviously, probably there are aspects which are of perception which are completely hardwired. And I'm wondering about the crossover between those things in the middle. What parts of, for instance, the actual experience of watching a film and the visual technology of perceiving how a film narrative is put together through montage is a learned behavior and a learned aspect of perception, and what part is actually hardwired?
Wow. So when I talked about learned modules, I meant learned in a neurobiological sense of has to happen before you're 7 years old. The difference between learning to speak a language without an accent before puberty, and then you can also learn a language as an adult, but you never learn it in the same way. And you're talking about...
Well, we know that, for instance, people who watched films in the early 20th century when they first saw them did not understand montage the way that we do. But also that montage was created in different ways in the early 20th century because it's a language. I'm wondering to what extent it's a language which relates to completely conventional modes of seeing and interpreting, which are actually intellectual and not visual, and to what extent it's a language which is actually a part of a perceptive apparatus which is learned by brains.
I don't know, because I only know about how we've gotten better at, filmmakers have gotten better and better at cutting.
Yeah.
But I don't think that's something we've learned. I think it's something they've learned to do. They've
That's
learned to
going on,
take
too.
advantage empirically of the way our brains work. And of course there are conventions in film, but
Obviously.
and a language. I don't know. You're beyond me. I just work on low-level vision.
Hi, this is a wonderful lecture. Thank you very much. As an artist, I'm really interested in time and attention, and sort of tie together movies and baseball, again, with art. I'm interested in either your low-level research or a more higher-level experience about how time passes with the baseball players. They'll say they'll see the threads or the stitches on a baseball, and how at moments of great trauma or such, our vision, our mental vision, our physical vision changes time. And I'm wondering what that has
David
to do.
Eagleman did an experiment to address that question, and he had people bungee jumping. And it turns out that they didn't expand their time sense in any way. What you really simply do is remember more things. So your brain is wired up to learn about the environment, and if you get reinforced, you have these reward circuits that sprinkle out reward hormones and fear hormones and excitement hormones, right? And make things easier to remember. And if you get really excited, you remember those things. You just remember more things that happened at that time. So it seems like it was longer because you remember more things. It's like the moon illusion. The moon seems bigger on the horizon because there's more things around to compare it to.
Yeah, thank you for your wonderful lecture. It brings up a lot of thoughts of in the art movement, art history. I mean, the famous Magritte's, this ceci n'est pas une pipe, this is not a pipe. Of course, it's a perception and an image. What I was reminded of, or was interested in hearing, it's a whole nother lecture, I understand that, but the relationship between what you talked about, the neurological and the different tricks that our brain does and the reafferents on the retina and this type of thing, as opposed to what Desmond Morris talks about in his Biology of the Art. Now, I know he's much benighted and over the top at times, but he does say interesting things about what one looks for in a work of art, including, and he lists 5 or 6 items or elements such as balance, symmetry, which aren't the same things, and optimum heterogeneity, departing from balance to the degree that the being out of sync is just right. And an artist who may be dyslexic or an artist who is colorblind, they still will they not be subject to these artistic talents or gifts for just doing the right thing out of sync. And I'll leave you with a quote that when he was asked about his egg-shaped things, the Brancusi, the perfect shape, he said, what do you do? He said, I look at it, I try to make it not a perfect circle, but he said, I know just when to stop because it's the excrescence that is the art, the imperfection that is the art. So is there any place for your studies to bring in the baseball player? That's a good analogy, the crack of the bat. They say the outfielder turns and runs to where it's going to go when he hears he can put together these sound, he can put together the eye coordination. The goalkeeper can stop if he sees the foot of the kicker. He knows which side to jump. But isn't there a similar group of things in art for the artist that has, regardless of all the physical or neural disabilities?
I'm not sure what the question was, but yes, I agree that artists are experimentalists and they do it until it hits them right.
I was really interested in what you said about amblyopia, which
I had when I was a kid. I am an artist. And I was always told that my eye could not see properly. What was used, letting the other eye see for it. So my eye was patched when I was 8 years old. And I remember what it was like to see through the bad eye with glasses. It was very cloudy trying to see the board, but within a few days it got clearer. And I'm trying to connect that with what you were saying about amblyopia.
So when you're young, usually before 8, but maybe there's still some plasticity, the brains of children are more plastic than the brains of adults. So if the brain is getting misaligned inputs, it will, one of its options is to ignore the input from the other eye. And when it does that, these connections being ignored just wither away. And so if you then close this, it's a competitive mechanism. So if you close this eye, the good eye, if you're still young enough, you can get some of the connections back from the bad eye. That's what they were doing. You were lucky you were patched, and it worked even a little.
Yeah, and I have a husband who's also an artist who had an operation for his eyes angling out, and he used to look through those stereo-opticons. That was part of the exercise that he was given to help his eyes. Just very interesting. Thank you.
Hi, thanks for such an interesting talk. Concerning your hypothesis about how equal luminance and color can lead to this shimmering effect in paintings, and I was wondering, do you know...
Joseph Alper's thesis originally.
Oh, okay.
I get all this stuff from artists. I don't.
Okay. Do you know if the colorblind or dichromats have suffer less from magazine ads like this?
Yes, because their equal luminance point will be different. But if someone would bother to make magazine ads just for dichromats, they would find them just as disturbing. But no one makes ads for colorblind people.
Hello. Are our eyes currently evolving?
Are we?
Yeah.
We're selecting for something.
No, our eyes at this point in human history, are they evolving?
Are we evolving? I'm trying to answer your question. Of course, there are differential survival rates for certain characteristics, but we're at a point in our society where we tend not to have selection pressure for good vision. Evolve. You mean evolving?
Yeah.
In the Darwinian sense?
Yeah, exactly. Because they're being
We
used.
have a pretty long, I'm sure we're evolving, but I don't know what we're selecting for these days.
Yeah, but we're changing so much that what we look at.
No, right, but you don't, evolution doesn't work on what you look at. Evolution works on whether or not you have babies. And if what you look at and are good at looking at
Well, if you pour acid,
is related to whether or not you have babies, then yeah.
Yeah, but let's go about it this way then. If acid is poured on flesh, scar tissue comes on the flesh to protect the flesh, the surrounding area of the flesh. If the eyes are subjected to being used in radically different ways from what they have been prior to a point in time, do they not tend to make up for a different usage or structure themselves in a different way for a different usage?
Only if it makes a difference in how many babies you have.
Huh.
That's the way it works.
Having no children, I guess that explains it to me.
I think maybe we have time for one last question over here.
This is another baseball observation that may be of interest to you. Mickey Mantle, you had his picture up there on the screen. One time was, and this is a documented observation, one time was out drinking heavily in the evening, and he came in, Casey Stengel put him on the bench because he couldn't even walk. He was so inebriated. But in the bottom of the ninth inning on a tie game, he needed a pinch hitter, and he brought him to bat. He barely made it to the home plate. And then when he got up there on the very first pitch, he hit the longest home run of his career outside over Yankee Stadium in the apartment house across the street. It was a tremendous shot, and never equaled that again. And when he came back into the dugout, the players asked him, said, "You could barely walk. You were so drunk. How did you do that?" He said, "Well, I saw 3 balls coming to me. I saw 3 balls, and I swung at the one in the middle."
Thank you.